All right, so we're almost done. We're almost done with cellular respiration, but we do have the last of the sub pathways, which is a fair amount of information. But let's try to get through the electron transport chain. So again, this is the third and final of the sub pathways of the basically the cellular respiration. So we started with glycolysis to cellular respiration, period oxidation, Krebs cycle, now to the electron transport chain. So the final sub pathway in cellular respiration. Right? So almost there, right? So we know we spent a lot of time making NADHs, making these electron transporters. Now they're going to have an important role to bring electrons to this electron transport chain. So let's start off first with the physical location. Where does the electron transport chain happen? So it occurs on the mitochondrial inner membrane, or the inner mitochondrial membrane. So if we look at this picture over here, so we have here cytoplasm. That first black line would be the outer mitochondrial membrane. And then in orange, we have the cristae. So this orange would represent the cristae. Another black line, the inner mitochondrial membrane, which would then be this membrane here. And then yellow, we have then the matrix. So matrix, inner mitochondrial membrane, and cristae. So that's sort of our physical location. So where does this take place? It's the membrane between the cristae and the matrix. So, and if you notice here, it's our phospholipid bilayer. We have our proteins, we have our sort of IMPs. So that is what the actual, I should say that's what the actual ETC, electron transport chain, is comprised of. And I'll show you a better picture in a bit. Now, when we talk about the electron transport chain, I'd like for you to think of it in the terms of a hot potato game. Right? So I don't know if we play hot potato, little kids party. You get a little animal or toy, or you get it and you pass it, and you get it and you pass it, and you get it and you pass it, right? I don't remember ever playing with an actual hot potato, little potato that can burn your hand. I don't remember ever playing with that, but maybe some of you did. Right? So if instead of a potato, we think of electrons, we'd go back to that idea of redox reactions, reduction, oxidation reactions. So one molecule gets the electron, it gets reduced, turns around, passes it, undergoes oxidation. We're going to make a simple kids game. We're going to make it all nerdy now by putting high-tech vocabulary on there, right? Oxidation and reduction. So reduction, gaining, followed by oxidation, giving up. And that's the process of the electron transport chain. So understand that each time we're shifting electrons like this, we're giving up a little bit of energy, a little bit of energy, a little bit of energy. So if I'm standing on this step and I want to go to the floor, I have two options, right? I can go this way and go down multiple steps, or I can take one step and go this way. The same amount of energy will be expended to get to the ground level, except this is going to release energy in more controlled, little bit of energy, little bit of energy, little bit of energy. So it's not going to cause any injury or harm when we get to this point. If I release all the energy in one step, it's going to get me there faster, but I'm going to splatter when I get to the ground. So if you think of a bomb, a firework, it's a tremendous amount of energy released in one violent reaction, explosion. Right? If that happened in the human body when we ate glucose and we start to break it in one reaction, it would explode and generate too much heat to be too destructive. So in order for it to be safely metabolized, it has to go through all of the steps in period oxidation, first all the steps in glycolysis, all the steps in period oxidation, all the steps of the Krebs, and now all the steps of the electron transport chain. So it's a lot for students to remember, but it's good because it's releasing energy and converting into ATP in a very, very safe process, right? Safe for our body. And even still, it generates heat. It generates enough heat to keep us at 98 degrees Fahrenheit, which is 98.6 degrees. That's pretty hot. If you go outside on a day that's 98 degrees, it's hot, right? That's a lot of heat, and that's what basically sustains our normal body temperature. So again, coming back to the sort of the anatomy of our electron transport chain, let's kind of get a visual of where we are here. So from inside out, we're in our mitochondrial matrix, our inner mitochondrial membrane, our cristae, and then the outer mitochondrial membrane. And this space up here would be cytoplasm. So cytoplasm, outer mitochondrial membrane, cristae, inner mitochondrial membrane, and matrix. And where does the electron transport chain happens? Or where does it happen? It happens in the inner mitochondrial membrane. So these are our characters that comprise the electron transport chain. So these are our characters, and I hope you notice here, NADH reductase, oxidase. So what are these? They're basically IMPs that are serving the role of enzymes or enzymatic processes. They're going to have active sites. They're going to interact with substrate. So these are important characters in the inner mitochondrial membrane. Now, in order to begin our story, we have to have an electron transporter. We have to bring the hot potato. We have to bring the electrons. And that's going to be brought in by our electron transporter, NADH. So NADH undergoes oxidation to form NAD+. And this electron is then given to our first participant in the electron transport chain. So this is an important starting point, right? Because without NADH, we can't then have the correct flow of electrons through here. So when this process happens, we're going to now start to notice two types of movements, right? We're going to notice the movement of electrons kind of sort of through the membrane, as they're going maybe from left to right through the membrane in this case. They're going within, and then going across the membrane are going to be the hydrogen cations. So the kinetic energy of the flowing electrons is then going to be used to pump hydrogens from matrix to cristae. So it's an important process there, right? And I mentioned the secondary transporter that's not as efficient, doesn't go quite to the right spot. That's FADH2. So it arrives late and in the wrong location, but is still able to contribute electrons to our system here. So it's contributing electrons. FADH2 undergoes oxidation, reduces then ubiquinone, and we continue on that process. And flowing electrons through the membrane are able then to pump hydrogens across the membrane from matrix to cristae. And this continues and it continues and it continues as long as electrons are able to flow. If the flow of electrons becomes damaged or stopped, we don't have an efficient movement here, right? The flow stops. Like, we can't allow that to happen. So we need a final electron acceptor. We need something with very strong affinity of electrons to pull the electron out of the ETC, the electron transport chain. We do have such a molecule, and that molecule then is going to be oxygen. We've talked about oxygen before. Chapter 2, we said oxygen was very strongly electronegative, has a very, very strong attractive force of electrons. So if you're an electron, would you rather be with oxygen or you'd rather be with this enzyme? It's not a fair fight. Oxygen is too strongly electronegative, has such a high affinity for electrons that it pulls the electron out. So oxygen pulls the electrons out and interacts with some hydrogens in that matrix to form a very safe molecule that we know of as water, H2O. So again, this only happens in aerobic conditions. If we don't have oxygen, we don't have that ability to pull electrons out of the ETC, and it will clog up everything. So we need aerobic conditions, and this is specifically the location where oxygen has its impact there. Yep. So as long as oxygen is there, electrons can flow. We continue bringing electrons, hot potato game, oxidation, reduction, oxidation, reduction, oxidation, reduction, oxidation, reduction, oxidation, reduction, until the final reductive event happens. We oxidize the oxidase, and we go then to oxygen. So this will continue, and we continue pumping hydrogens across that membrane. And we then basically have a strong acidity that starts to generate. So we're starting to build acidity up in this cristae, and we have a strong concentration gradient. So again, concepts from chapter 6 now, right? Chapter 2, acidity, and then chapter 6 now are concentration gradients. And what happens when we have a strong concentration gradient, right? What happens with this? Is the cell going to be happy to leave it like that? No. So eventually these hydrogens are going to have to diffuse back to the matrix. Come back to this. And they're going to diffuse back through a very important enzyme, arguably the most important enzyme in all of biology, which is what we call ATP synthase. So ATP synthase, that's the name of this enzyme. It will allow the movement of hydrogens from high concentration down to low concentration. So the diffusion of hydrogen cations. And when that happens, we call that chemiosmosis. That's the name of this process. So chemiosmosis, the movement of hydrogens to make a concentration gradient, and then the diffusion of those hydrogen ions to work back to a state of equilibrium. Chemiosmosis. So ATP synthase, think of it like a little valve that right now is closed. When we get to the right concentration gradient, that valve will open and allow then hydrogens to flow from high concentration to low concentration. And it will kind of spin this thing like a turbine. And in that process, it gives us enough kinetic energy to load a phosphate to ADP to form then ATP. So I'm simplifying here a little bit. It's an intricate process, but very critical for the formation of about 32 ATPs in humans. So again, same concept, just very, very simplified here. So how does the electron transport chain work? Right? First, we need our NADH to give up its electron. Electrons will flow through the membrane, and hydrogen ions will flow across from matrix to cristae. We build up our gradient of hydrogens, and then they're going to diffuse back through this ATP synthase enzyme to help generate then ATP. So again, it's not that it's that super difficult. It's just a lot of things to remember. It's very tedious, right? Step by step by step by step. So just again, spend some time with this. Try to make sense of as much as you can. I like this picture because it throws everything at once at you, right? If you can make sense of this picture, there's nothing I can ask you that on the next test that you won't get, right? So definitely spend some time and effort analyzing this. Maybe if you get really confident, maybe cover the words and then see if you can fill in the words on your own, right? But let's start here, top left. So I'm in the cytoplasm and I have pyruvate. So you know what must have already happened. Which pathway must have already occurred if I already have pyruvate? Right? I hope you tell me that glycolysis should have happened, right? Glycolysis, and that's correct. Glycolysis must have already happened for us to have pyruvate. So pyruvate is formed. Pyruvate goes from the cytoplasm. Well, first I'm sorry, this is not cytoplasm, this is the cristae, right? So we formed pyruvate in the cytoplasm, went through the outer mitochondrial membrane, now into the cristae. So we're in the cristae here. And we diffuse into the inner mitochondrial membrane into the matrix. So here's our inner mitochondrial membrane. There's our electron transport chain. Okay. But first, we have to go through pyruvate oxidation. So we generate our NADH molecules. We form our acetyl-CoA. Our acetyl-CoA then allows us to enter into the Krebs cycle, where we make more NADH, 6, 2 FADH2s, 4 more CO2s, and 2 ATPs by the process of substrate level phosphorylation. Now, all these NADHs then move to the electron transport chain. They give up their electrons in that redox chain, that hot potato game. Electrons are going to flow through the membrane, while hydrogens are moved across the membrane back to the cristae. We want to keep this electron transport chain going, so we need our very strongly electronegative element, which is oxygen, to pull the electrons out of the ETC. They're going to combine with hydrogens that are in the system to form water. Right? All the hydrogens then start to diffuse back through that channel protein that we know of as ATP synthase, and that process generating 32 ATPs. So again, a lot of stuff happening simultaneously, but we do know it is a sequential type of process. Right? But again, lots of stuff, but I have confidence in your analytical abilities to kind of put all this together there. So let me back up. So this is an average. This is an average. It gives us some theoretical energy yields. So on an average situation, right? If we look at NADH dropped its electrons off farther from the endpoint. So its electrons traveled farther, therefore pumped more hydrogens up into the cristae. FADH2s didn't travel as far, therefore didn't send that many hydrogens into the cristae. So we know that the more hydrogens are there in cristae, the more ATP can be formed in that ATP synthase chemiosmotic process. So a rough estimate, and again, this is an average, it's not exact. Each NADH is able to generate about 3 ATPs per molecule. So for every 1 NADH, if everything works correctly, it should generate 3 ATPs at the end through ATP synthase. Each FADH2 is only able to generate 2 ATPs per molecule. So not as efficient, but still ATPs, yeah. But again, we're gonna come back into those numbers here. Forward again, forward. All right, so just again, trying to zoom out and show you everything, starting from glycolysis, which formed pyruvate. If there was no oxygen, we stay out in the cytoplasm to undergo fermentation to produce lactate. End of the story. If there was oxygen, pyruvate enters into the mitochondria, where we go pyruvate oxidation to form acetyl-CoA, Krebs cycle then to form more NADH, FADH2, 2 ATPs by the process of substrate level phosphorylation, and garbage carbon dioxide. NADH then travels to the electron transport chain to generate 32 ATPs. We also form water. Right? So the summary reaction, which seems very simplified now, I hope you see. We started with C6H12O6, glucose way over here in glycolysis. We know that we have 6 oxygens that were utilized way over here at the end in the electron transport chain. We formed a total of 6 carbon dioxides. We did 2 from the pyruvate oxidation and then 4 from Krebs. 6 waters were all formed in the electron transport chain, and we form an average of 36 ATPs, right? So this is where it's an average again. It's not an exact thing. It's going to depend on person to person. It's going to depend on athletic training, how many total mitochondria, how effective, how efficient those mitochondria are. But again, if we add up the math here, so we got 2 ATPs by substrate level phosphorylation in glycolysis, and the same way that we did in the Krebs. So 2 and 2, those are exact numbers, right? Those are, you know, +2, +2. And we formed 4, but 2 of them were invested. So a net profit of 2. In basically this process in the Krebs, it's about, right? 0 to about 2. So it's not exact, right? And then in the ETC, about 34. But for most people, it's going to be 32. So we have the potential to generate more ATP, but on average, we generate, say, 32 plus 2 plus 2. So it gives us that 36 number. It can be lower, and I know some individuals, they don't get 32, they get about 28 only from the ETC. So it just, those are average numbers, just like everybody has a slightly different normal body temperature. Some of you are 98.6, some of you are 98.4, some of you are 99 and you're perfectly fine. It just depends on the individual. Right? But this is a pretty standard number. This is the number I'm going to ask on the exam. So 36 ATP, counting everything from glycolysis all the way to the end of the electron transport chain. So to summarize everything, so we started with glucose. We make 2 direct ATP through substrate level phosphorylation. We generate then the 2 NADHs. We send, this box is in the cytoplasm. This sort of separation indicates now we are in the mitochondria. So we start out with pivot oxidation, which gives us 2 NADHs. We go into the Krebs, where we have 2 direct ATP through substrate level phosphorylation. We have the 6 from NADH and the 2 from FADH2. So 6 times 3 gives us 18. 2 times 2 gives us 4. So everything looks good here. 2 times 3 is 6. 2 times 3 should be 6, but here's our little discrepancy. So we only get 4 ATPs from the 2 NADHs produced in glycolysis. Understand, the longer your Uber drive is, the longer you ride the taxi, the more they're going to charge. So all of these NADHs were found in the mitochondria. These NADHs are coming from the cytoplasm, so they have to travel farther. They lose energy in that process. So again, we have the potential to make 38, but on average, it's only 36 because of that inefficiency there. Again, that can be changed as well, can be made more efficient. But the number that I'm going to ask on the exam, again, 36 ATPs. So almost done, just about done, right? We covered glycolysis all the way through, right? But this is that's that overall sort of structure, the skeleton of reactions there. But this is only for carbohydrates. And carbohydrates are the easiest molecules to metabolize. So if the body still needs energy and we run out of carbs, we then have to shift to plan B, which are the lipids. And we don't call it glycolysis, we have to call that lipidolysis. It's a whole other set of enzymes, a whole other set of reactions, another headache. If I had another 4 hours to talk about lipidolysis, I would cover that. And then if I had another 6 hours after that, I would cover proteinolysis, basically. Proteins are even more complicated. They're more structurally sound, so they're harder to break apart for energy. So your body will always prefer the simple task. If the simple task is not there, we go to lipids. And if lipids are gone, we got to go to the proteins to metabolize those for energy. So again, carbs, lipids are broken throughout their own pathways and fed into then this sort of structure of metabolism. Proteins as well can be introduced into different parts of that overall molecule or that overall pathway. But again, that's for your Um, that's for your basically biochemistry class later that you got to take that. I'm not going to get into that now. So just trying to show you, give you like a timeline and a realistic ideal of this. So we look at here the 3 categories of macromolecules, and we have some starving child here. If we look at this first little red line, that is basically carbohydrate energy. The carbohydrate macromolecules will sustain this child for maybe about, this is 1 week, maybe about 2 days. So not a lot of energy held in carbs. Very easily metabolized energy, but not good for long term. We say that's a short-term energy. This next blue line is going to sustain. So this is plan B. This is now your lipids, which can sustain for weeks, right? So instead of days, now we're talking about weeks and weeks and weeks and weeks. Once we start to deprive the body of lipids, then we shift to metabolism of proteins. So again, if the body is relying on proteins to stay alive, basically that's a very malnutrition, very starvated state of physiology. So the body starts to metabolize muscle tissue, connective tissue. It's just, again, not a good scenario, right? But keep you alive. And I hope you understand there's no 4th line. We do have 4 macromolecules, but nucleic acids are not found in this. There's a lot of phosphates in nucleic acids, but the body, the cell is not going to metabolize the energy. The boss is not going to get metabolized for energy. It's going to send out everything else. The cell would rather die than actually metabolize its nucleic acids. Kind of strange and weird, but that's the way that it has evolved. Right? So with that, it's a lot for you to basically to soak in. Take you a little while. You have the weekend now, and you have next week to get through this, but I wanted to upload all of this. I'll upload a few accessory videos that should help out to visualize everything there. But with that, it's a lot. It's a full day, a lot for you to go over. But with that, I'll call it a day, and I'll be seeing you next time. So y'all have a great one.